This is lecture 6 in the course Principles of Metabolism. In this lecture, we will look at isotope tracing methods to study metabolism. So the principle of isotope tracing is really very simple, and I'm sure most people have seen this in some form or another. So what you do in isotope tracing is that you give a cell culture or a tissue or even an animal a substrate which is labeled in some position, perhaps on a carbon. And then you let the cells or the tissue process this for a while, and then you recover products and you check what is labeled. And of course, what must happen is that if you discover a labeled product, it must have come from the labeled substrate or the tracer. So here is an example shown. If you put in labeled glutamine to cells and you recover labeled lactate, then you know that there must have been a pathway between these two. This particular pathway is called glutaminolysis. So you don't know anything about what that pathway actually would look like. You can go to biochemistry books, but the experiment doesn't tell you that. But you can say that it is there is a pathway, and it's turned on in these conditions. So that's pretty simple. So to do this, what you need to have is you need to have some kind of recognizable label, some way to label a molecule so that you can recognize it in products afterwards. And you need to be able to label it in a way so that it doesn't interfere, so the label itself doesn't interfere with the metabolic pathway. Otherwise, you will get artificial results, of course. And so for this, many isotopes are available, and most of them are quite neutral. They don't interfere with metabolism themselves. But they are easy to detect. So isotope tracing has been used for a long time in the study of metabolism, and it has actually been used to map out many of the metabolic pathways that we now know. So this is an example from 1961 from Melvin Calvin and his work on the photosynthesis cycle, so the light-independent reactions. So Calvin used 14C labeled carbon, which he fed to photosynthetic bacteria for some period of time. And then he took extracts from these cells and he ran them on a thin film chromatography method. So what you see here is a two-dimensional chromatogram, and these dark spots are the various metabolites that were labeled from 14C. So in this way, he could study products that were formed by the photosynthesis process. And what he went on to do is to repeat these experiments at different time points. So he did detailed time series, and so he could see which products were formed first and which were second, and so on. So he could come up with a sequence, and in that way he was able to reconstruct this cycle that we now know as photosynthesis. And so in many ways, we still do isotope tracing in the same way that Calvin did, but we have, of course, more refined methods of measurement, and we have better accuracy. We can study more things. And we also now know a lot about what the biochemical pathways look like. And mostly we are nowadays using isotope tracing to measure pathway activity. We kind of know what pathways should be there, not all of them, but many of them. But we're interested in their activity in different cell types or conditions. So this is an example, a more recent example from 2011 in a paper where the authors studied glutamine metabolism in the TCA cycle, and specifically the use of glutamine to make citrate for fatty acid synthesis. So this was a mode of metabolism that was called reductive carboxylation, and the whole idea of this, the whole finding of the paper, is that glutamine has a pathway where it can go sort of backwards in the TCA cycle and form citrate. And this goes on to form fatty acids. And the way they establish this is by cleverly choosing a label, a positioning glutamine where it's labeled, so that you could say in an unambiguous, clear way that this really goes to citrate, and you can prove that. So here, the trick is really to choose the label, the tracer, in a good way. So what you should choose here, if you want to show that glutamine goes to citrate, you should pick carbon 1. And the reason you should do that is because if glutamine goes into the TCA cycle and starts turning, then carbon 1 gets lost immediately. So whatever happens to the rest of glutamine in the TCA cycle, it can be very complicated. You see these other atoms are rearranging in various ways. But we don't have to care about that, because this carbon is always lost. So the only way this carbon 1 can end up in citrate is actually if it takes this route. If it goes backwards and then forms citrate this way. So you can go through the biochemical reactions, look in detail at these steps and make sure that that's correct. And when you're sure of that, then you have a method for determining whether glutamine goes to citrate. And this is typical in designing tracing experiments. You have to understand the biochemistry and exactly how these molecules are rearranged. And then you have to figure out a way where you can see that if this product is formed, if citrate label is formed from this tracer, then I can be certain that my pathway of interest is actually active. And so that's what they did in this paper. And that's a typical use for isotope tracing to study activities of pathways. So a few concepts which are useful to know about when studying isotope tracing. So let's say that we have labeled a cell culture with carbon-13, and we are looking at the possible ways that this carbon-13 can get into a 3-carbon compound like lactate. So then you come up with a number of possibilities. So here we draw compounds like this, just 3 circles for 3 carbon atoms. And if we have a carbon-13, we draw a black circle, otherwise it's open. And so you can see that there are, in this case, 8 possibilities in which you could get labels into different positions of lactate. And these are called isotopomers. So this is the same metabolite, but it exists in 8 different kind of parallel states with different labeling patterns. That's isotopomers. Now, it would be great if we could measure all of these and distinguish between them, because that would give us a lot of information about metabolism and how this compound is formed. Usually we can't do that. So it depends a bit on how we measure lactate, what measurement technique we use. But a very common way of measuring these isotopomers is mass spectrometry. And so in mass spectrometry with stable isotopes, what we can do is we can distinguish between things that have different mass. I mean, that's mass spectrometry. And as you can see, these group of 3 isotopomers all have 1 carbon-13 in them, so they have the same mass. They are exactly 1 carbon-13 heavier than the unlabeled compound. So we call this +1. The unlabeled lactate we call 0. And then there's a group of these 3 patterns. It's a group of these 3 patterns which have +2 carbon-13s in them. So those all have the same mass. And then finally there is the +3, the species where all the 3 carbon atoms are labeled as carbon-13. So if we measure this by mass spectrometry, we'll only be able to see difference between masses, and this is called mass isotopomers. So we can think of this as groups of isotopomers that have the same mass. Now, if we consider that in our actual cells, we have some of these species appear, some don't, and some are more common than others, depending on what metabolism looks like. You can come up with a distribution over these. So these are basically fractions of the total. So you might have 20% unlabeled, you might have 10% of this, 40% of this particular form, and so on, adding up to 100%. But if you look at this as mass isotopomers, then of course you're not able to distinguish between these 3, for example. So all you'll be able to say is that as a total, the +1 heavy things constitute 10%. And similarly, these 3 particular species, this is actually might, for example, be the most common one, 40%, but you can't tell that. All you can say that in total in this group, there is 50%. So you can measure the abundance of particular mass isotopomers with mass spectrometry, but you can't actually measure this. But that gives you some information, although it is partial. But we will see that this sometimes limits what we can do. It has one nice sort of side effect, though, and that's that if you look at isotopomers, there are usually very many of them. So if you have large molecules, the number of possible patterns grows exponentially. So if you have, in extreme case, a large metabolite like cholesterol, you can have hundreds of millions of isotopomers. But of course, there's only n plus 1 possible cases. There is the unlabeled, and then you have plus 1, plus 2, up to plus 27. So these correspond to large groups of patterns, but you only have 27 of those. So the data is more manageable, but contains less information. So let's look at some examples of how this type of species, isotopomers, can form in metabolic pathways, and what happens to mass isotopomers and the stuff you actually can measure. So here's an example, a couple of enzymes from the TCA cycle. So let's say that you are able to label your cells with pyruvate, where you have put in a carbon-13 in this position. Now, let's say you observe citrate, and you observe that it has one carbon-13 in it. You don't actually know where it is, but you observe that there is something in it. So one possibility is then that pyruvate might have entered into acetyl-CoA, keeping this carbon atom which is labeled, and then it has condensed with oxaloacetate. So this is the first sort of step of the TCA cycle, and formed citrate. And so therefore you will have a plus 1 because this carbon atom will be retained. That's a possibility. On the other hand, there is also another possibility. There is an enzyme that takes pyruvate, adds one carbon atom, one carbon dioxide, and makes oxaloacetate that way. And so it could be that the acetyl-CoA is actually unlabeled, and your carbon-13 comes in this way instead. And so maybe you actually had a citrate molecule that looked like this. So this is, of course, different from what we had up here, if you actually were able to see the position, but they all have plus 1 weight. They all have one carbon-13 atom in them. So if you measure this by mass spectrometry, they would be indistinguishable. And these two pathways would actually tell you something different. They would mean a different metabolic state. This would mean perhaps glucose oxidation in the TCA cycle, but this might mean that you actually get your substrate from somewhere else, maybe fatty acids. So in this case, you might have a situation where you are not able to distinguish between the pathways if you use mass spectrometry. And then you could have more complicated situations. So let's say, for example, that both of these pathways that we looked at could be active. So maybe there is some coming in from pyruvate this way, and there's some substrate coming in this way. And then maybe you also have unlabeled sources of this. Maybe you could get unlabeled oxaloacetate, unlabeled acetyl-CoA. And now you start to get these kind of combinations and lots of isotopomers. So one possibility is that the one we had up here, that you had labeled acetyl-CoA and unlabeled oxaloacetate. But you could also have both of these labeled species condensed in this reaction, and then you would get this, which is plus 2. This you would actually be able to distinguish by mass spectrometry then, because it has 2 carbon-13 atoms in them. These are the two cases we considered previously. And then in this case, there's also a possibility that you get a completely unlabeled citrate. So this is examples of what could happen. In this case, if you were to see a plus 2, what can we say? Well, then we know that pyruvate has entered this way because there has to have been one labeled carbon up here. And we also know that it has to have entered this way. So in that case, we would be able to say that both of these ways, these enzymes were active. If we saw a plus 1, well, then we're back to the previous case. Then either of these routes must be active, but we can't really tell which one. So that's an example case that could occur. So isotope tracing is a really powerful method. It's really the only way in which you can monitor the activity of enzymes inside living cells. So that's pretty special. But it has its limitations, like everything. And depending on how you design your experiment, you will be able to see more or less. So it's important to know how to do this. So here's an example where if you design the experiment correctly, you might be able to see something and otherwise not. So here we are looking at glutamate, and we are looking at reactions which convert glutamate into alpha-ketoglutarate. So this is a way of removing one amine group and exchanging it for just a keto group. So if you look through databases, you'll see that there are at least two ways of doing this. There's a dehydrogenase, glutamate dehydrogenase, that simply removes the ammonia. And then there's a whole bunch of aminotransferases that take this group and transfer it to another amino acid, to synthesize amino acids. Both of them could yield alpha-ketoglutarate. So how can you tell what is what? Well, first of all, the entire carbon skeleton is intact from here to here. So there is no way of doing this by simply looking at carbon. So what you need to do is you need to label the ammonia group, the amine group. So the nitrogen is what's really changing. And so now you may or may not be able to distinguish between these two alternatives. And so it depends now on what you can measure. So this is definitely the right choice of tracer if you want to decide this. But are you able to measure all the products? So probably you'll be able to measure the amino acids. There are lots of methods for that. So you'll be able to say if particular aminotransferases are active. So that's good. But you may not be able to see the ammonia. This might be hard to measure with many methods. So, for example, you could end up in a situation where you can detect activities of these, but not this. And you have to choose the right tracer to be able to do that. So, in general, the situation is that what we are able to detect and what we're able to distinguish between, it depends on how you choose your tracer, so that has to be done with some care. And it also depends on what metabolites are measurable, where can you actually detect label. And these two things interact. So there are some systems that kind of support designing these experiments. Some of the databases that have maps between atoms to atoms in reactions can be used to ask what should I label if I want to have something from here that ends up here. So there are some systematic ways for doing this. In some cases, we are also able to say something about the relative rates at which a metabolite is being produced using stable isotopes. So not only detect that an enzyme activity occurs, but how large is it? So quantitative measurements of fluxes. So how is this done? Well, if you do an experiment where you can put in labeled pyruvate, for example, into cells, and you're able to measure the amino acid serine, and you know there is a pathway that can form serine from pyruvate, and you also know that there could be some uptake of the amino acid from the medium into the cells. Now, if you're able to measure the fractions of labeled serine, you can say something about what the relative contributions of the synthesis pathway from pyruvate is compared to the uptake of serine. So not absolute numbers, but relative. And the reasoning is fairly simple. So if this metabolic system is at steady state, so the fluxes are constant and these numbers are not changing over time, then you can say that since we observed 30% of this and we had a completely labeled pool here, well, then we actually must have 30% of the flux coming this way. The other 70% are sort of serine that has been diluted out with unlabeled serine from the surroundings. So it turns out that the proportions between the synthesizing enzymes, their activities, is the same as the proportions between the isotopomers. And this is pretty much the overall principle of trying to do flux analysis with isotopes. But there's a bit of a complication. So here we assume that we had fully labeled pyruvate. Normally that's not so easy to do in practice. So what if we have this situation that we're only able to get 40% labeled pyruvate into the cells? So we actually have an isotopomer distribution like this. And then we measure serine and it's 30% labeled. Now in this case, how much as a fraction of the serine is coming from the endogenous synthesis pathway from pyruvate? Well, the reasoning here is that 30% of label here, well, that's actually 3/4 of the 40% of label you have up here. So the way you determine the flux in this more general case is that you take the isotope fraction that you had in serine in the product, and you scale it by what you had in the substrate. And therefore, in this case, 75% of the total flux comes from pyruvate. So 75% of the serine that is synthesized comes this way, and only 1/4 comes this way. And that's a pretty big difference, 30 versus 75. So this matters, and we have to scale the results in this way for these calculations to be correct. Now, the general case, you can do this in more complicated networks. So if you have any product that you're interested in, and this can be any isotopomer of any metabolite that you can measure, if you have a number of possible reactions, n of these with fluxes that contribute to the product, and they come from these n substrates, then you can do this type of calculation again. And so now it looks like this. So this is the relative fraction that enzyme I is contributing to the pool, and that relative fraction is the same as the fraction between these isotopomers, this one, and whatever you have up here. So that's just the general case of what we did up here. And so you can do this for any metabolite, and so you can always determine the relative rates of enzymes that is producing something that you can measure. You can also view this in a second way. So you can view this as a mass balance sort of phenomenon. So if we just rewrite this equation slightly differently like this, you can say that what's actually happening here is that the amount of this isotopomer I that is produced by this reaction must be the same as the amount of this isotopomer that comes out from whatever reactions are consuming the product. So this is actually a kind of mass balance phenomenon, very similar to what we saw in the previous lecture on flux balance analysis. But here we have many more things we can measure. We can measure all these isotopes, isotopomers, and therefore we get much more information. And therefore, stable isotopes is very informative for figuring out the fluxes in cells. But there's a caveat here, and it has to do with the fact that when we talk about fluxes through metabolic reactions, there are actually several of them, as we said in the previous lecture. So there can be a forward flux in a reaction from A to B, but there can also be a reverse flux going on at the same time, so that you have some amount of material that is just exchanging back and forth, sort of in equilibrium, and then you might have a net flux from A to B on top of that. And what happens is that when you do isotope tracing, isotopomers will reflect the forward flux or the reverse flux, depending on where you put in the tracer. So if you put in tracer here, it's going to end up in B as long as there is some flux in this direction. So it can be that this reaction is actually at equilibrium and there is no net flux, or it can even be that it's going in reverse. As long as there is some forward flux in this direction, then you will see tracer coming out in B if you put it in here at A. So this can give some counterintuitive results when you look at isotope tracing studies. So if we go back to our example, as we had before, same thing, pyruvate synthesizing serine, but we now consider this step to be reversible. So what could happen? Well, the proportions of serine coming from pyruvate and from uptake from the environment will be the same. So it's still going to be 3 to 1, 75% of serine coming this way. And it could be that this reaction is actually irreversible and there is a net synthesis of this, so nothing going in this direction. But it's equally possible that you have something like this. You could have this reaction being completely at equilibrium, and there's actually just an exchange between serine and pyruvate, but there is no net synthesis. So when we say that from isotope tracing, we see that 75% of the serine derives from pyruvate, that can still actually mean that there may be no net synthesis. So this is this can be a counterintuitive and confusing concept. So make sure to look carefully at these examples and see that you understand this, because it becomes important when looking at the literature. So let's look at a particular case and revisit the study we mentioned before on reductive carboxylation. So in this case, we said that if you can see this label from carbon 1 of glutamine showing up in citrate, then you can prove that there is actually a flux in this direction. And that's totally true. But we don't know anything about the flux in the opposite direction. Well, this is the same problem. So it could be that there's actually no net synthesis in this direction of citrate coming from alpha-ketoglutarate. They could be at equilibrium. There could be nothing going on. Or it could be a net synthesis in the other direction, actually. You might still see a label coming out here. And this was pointed out in a follow-up paper by another group where they did some simulations and studied this. And they said that there's actually a wide range of fluxes possibilities at this reaction that is compatible with the data. And so just by observing the label in citrate, you can't conclusively say that there is a net synthesis along this pathway. So this is an important thing which is still being debated in the literature. So far we have looked at determining relative rates of reactions, relative fluxes. Now, if you also have some measurements of, say, uptake or release of substrates, then you can use this to kind of scale the system, and in that way you can also determine absolute fluxes through enzymes in living cells. So say that in this particular case we've been looking at, if you're able to actually measure the absolute uptake rate of serine, say that it's 10 femtomolar per cell an hour, and you also have this isotope data, which you can calculate and work out that 75% of the flux is coming in this direction, well, then you can simply scale the numbers. And by combining these types of data, you are then able to get absolute rates, in this case, of serine synthesis and of serine utilization in cells. And this is really the only way to get to these type of absolute quantification of metabolic activities in living cells. And it can be quite powerful. Now, as we have seen, these methods are quite complicated. It's very easy to get lost in little caveats and make mistakes with calculations and so on. Some people bravely do these type of calculations by hand, and you can find articles full of equations where you manually set up all these balances and fit it to data and draw conclusions. It can be done, but it's not easy. And so to support this type of work, there has been a development of systematic flux analysis methods. So these are automated to some extent. So there has been a series of papers in the early 2000s where these methods were formulated, and they have been improved in several generations, and they're now quite powerful. So these methods require you to have a metabolic network model. So these can be found from the databases we're working with, and a set of atom sort of maps. So which, in all the reactions, which atoms go where. So if you have this, and then you put in some tracers and you measure some isotopomers, then there are automated ways of taking care of all this stuff with equations, collecting them, combining these equations with uptake release data if you have that, and basically fit your data to a model. So this is not entirely straightforward. The software for this is still quite technical, and so you need to spend some time on doing this. But at least there are systematic methods and software that help you do this without having to deal with all the equations manually, because that can be really difficult. So these are a couple of papers that detail some systems for this. And you can find plenty of examples nowadays in the literature where this is being used. So this is one example. This is a really nice paper, I think, looking at using the systematic methods, looking at fluxes in the mitochondrion and in central metabolism in response to knockdown of the pyruvate carrier. So this is a way of seeing what happens to this branch of metabolism when you lose pyruvate entering the mitochondria. And this figure shows you some examples of fluxes that change, increase or decrease when you knock down this particular solute carrier. And there are several examples like this. So that's nice. So I think this field has gotten to the point where these methods are becoming quite powerful, and you can use them to really interrogate what happens with metabolic activities in living cells when you do these type of perturbations. But they still have limitations, and important limitations if you use this type of steady state flux analysis methods is that you will not be able to say anything about end products or linear pathways, so a sequence of enzymes that make some final product. And the reason for that is because, if you remember, we always need to look at ratios of reactions that produce a given metabolite. And if you have an end product and there's only one reaction that makes it, then there are no such things. So these things are not possible to get information about using these methods. For this, you have to either quantify it directly or you have to use some alternative means. So I'd briefly like to mention another kind of class of methods to do flux analysis, which you also encounter sometimes in the literature. So this is not based on steady state values in the same way. It's based on time series data, looking at how isotopes of a metabolite changes over time. It's a kind of, you might call it a pulse labeling type of experiment, where you have a system or a metabolite of interest which is unlabeled when you start the experiment, and then you start at time zero feeding in isotopes into this pool of metabolites. So you might, for example, be looking at lactate as the pool, and you might be feeding it isotopes by giving cells glucose, which is labeled. And what happens then in systems like this is that the amount of isotopes you see in your pool, which would be this, it behaves like an exponential type of decay. So you will have the unlabeled fraction will sort of wash out, and you will have the inverse curve of the labeled fraction actually going up. And so this type of system has some kinetics which can be described by a half time. So there's a characteristic time at which you reach 50% labeling in your system, for example. And you can calculate this according to this formula. Now, this is good in the sense that if you take some time series measurements of how much isotopes you actually have in this pool over time, then you can fit these equations to that type of data. And by using that, you can infer how fast is the flux going through this pool. So this is a different type of method. It still assumes that the metabolism is at steady state, so the actual size of this pool is not going to change over time, and the actual flux that's going through it is not going to change over time. But you will watch how the isotope changes over time, and from that you can draw conclusions. So this requires, it's a bit laborious. It requires you to make time series, so you have to make multiple measurements, and sometimes you have to do it at pretty tight time intervals, so that's not so easy. And if you are going to determine the fluxes, you actually, in absolute amounts, then you also have to know the pool size, so the absolute concentration of a metabolite inside a cell. So those are pretty difficult experiment and measurements to do, but it can be done. And if you can do it, it's a quite powerful method. And in some cases, you can use this to determine some fluxes which are difficult to get by other methods. So here's a couple of examples. So this is a paper from 2008 from Josh Rabinowitz's group in Nature Biotech, where they used this method to determine the fluxes in glycolysis and the TCA cycle and fatty acid synthesis. So this was a system where they looked at cells that were transfected with viruses or not, and they looked at what metabolic burden the cells have by having to produce virus particles. So what you see here is sizes of these fluxes, the relative activity, and also in colors you see how they change with this virus infection. So this is a pretty nice paper using this type of kinetic flux measurements. You also see variants of these methods sometimes in vivo studies. So people who look at fluxes between different pools of lipids or cholesterols or glucose or things like that in plasma, for example, sometimes use this type of methods. And it's usually because it's difficult to reach steady state, and you have the type of activities which are difficult to get by steady state methods. So here you typically infuse label into the plasma of a subject at a given time, and then you watch how the isotopes change over time, and from that you can draw conclusions. If you look at these more in vivo type of papers, like this one by Turner 2012, you will notice that the terminology is very different, and it doesn't kind of seem like the same thing, but it's actually the same principles that are underlying these types of studies.